Hey, everybody. So my name is Jeffrey Koury. I'm currently one of the editors at ResearchHub in the Neuroscience Hub. Although a lot of my work is also immunology, which you'll see in the presentation. And I'm also one of the leads at the ResearchHub Foundation. And so I wanted to talk to you all a little bit about some of my research, which primarily focuses on the role of HIV. Um, and it's in the context of the brain and some of the information we see there, which is a little bit lesser known, some of the neurocognitive deficits around that. And so I'll be focused primarily on talking about a protein, Ephrin-B1, which is a cell membrane protein, and also this interferon beta, which is a type 1 interferon, and the role of that in the innate immune response around viruses. So here's a little schematic of what I'll talk about. And this is actually kind of encompassing mostly my PhD dissertation. So just talk about HIV in hand, a little bit on the background and some of the mouse models we use, as well as some background and prelim data around what our lab's done on interferon beta in the past. And then some of the early things that I found that kickstarted this project around the Ephrin system. And as well as looking at a couple questions, which is what even regulates the ephrins in the brain, which was a question that was unanswered before this, as well as the focus on microglia and the ephrin system on the microglia, what's it doing in terms of inflammation and what might it be doing in terms of synaptic pruning. So real quickly, so according to the UN AIDS in 2019, there's about 38 million people living with HIV worldwide. And you can see it's a huge issue in especially the southern regions of Africa, but it's also like a fairly large issue here in the US. So in the US, about 1.2 million people living with HIV. And some of these stats may not be so surprising, but what really does surprise a lot of people is this prevalence of HAND, which is an acronym was stands for HIV-associated neurocognitive disorder, which most people don't attribute HIV to. And some studies have shown quite a range, but up to 55% of people with HIV infection have some level of neurocognitive deficits that accompany that. So you can see here in this schematic that you have various forms of HAND, and you have what's called the pre-CART and the CART era. CART is the combination antiretroviral therapies. So HAND is like symptomatically characterized by impaired memory, attention, and concentration, as well as like some motor deficits, like slowed movement. And you can see here from highest severity to lowest severity, you have HIV-associated dementia, mild neurocognitive impairment, and asymptomatic neurocognitive impairment. And I think what most people appreciated, and what was, I think, really like a really wonderful, impactful discovery was this creation of the combination antiretroviral therapies. And so you can see that the life expectancy has increased to that of just the general population. But what it hasn't done is decrease the prevalence of the neurocognitive deficits that accompany HIV. You can see in the pre-CART era, there was mostly the more severe form, the more full-fledged dementia-like symptoms. And in the post-CART era, you could see that the more severe form has decreased, but the prevalence of the more milder forms are still apparent, and thus the overall prevalence hasn't changed. So the current model for how HIV actually induces neuronal damage is you have the virus. The virus infects what most people know as CD4 T cells, but actually macrophages and microglia also express CD4, but at a lower expression. And so they're also a particular target for the virus. So they go in and they'll bind the CD4 receptor, but they also use this chemokine co-receptor. So these can be either CXCR4 or CCR5. This gives the HIV, there's different tropisms. So there's some that are more leaning towards using CXCR4 and some more leaning towards using CCR5. But ultimately what this does is it activates the microglia. And the microglia in turn will release various neurotoxins, as well as different viral proteins from the HIV virus that can damage the neurons. And additionally, you have a pretty rampant like cytokine storm, and those cytokines can also damage the neurons, but also result in astrocytosis. So activation of the astrocytes, which in turn will release cytokines of themselves as well as other reactive oxygen species that damage the neurons. So that's one way. And another way is the HIV virus can actually shed this viral envelope protein called GP120. You can almost think of it, it's very akin to the spike protein on SARS-CoV-2. And what this does is it binds to the appropriate receptors because the binding on for these receptors occurs via this viral envelope protein. But you don't have any of the other virus in there to infect the cell. But just that viral protein alone can activate the macrophages in the microglia. And you don't release viral proteins, but you do release neurotoxins that can damage the neurons and result in a lot of the neurocognitive deficits that you end up seeing. And then lastly, to mention that all these cells express these chemokine co-receptors, CCR5 and CXCR4, but it's only the microglia that express CD4 and can be productively infected by the virus, which is why in my studies, I'm particularly focusing on this as this is like the main kind of pivotal step in all the downstream behaviors of astrocytes in the neuron. So as I mentioned, there's this viral envelope protein, GP120, on the surface here. And we actually leverage that in a transgenic mouse model. And this is a well-characterized model from back in 1994, where they just essentially plug in the GP120 viral envelope protein under a GFAP promoter. GFAP is a common marker that's associated with astrocytes, which are very exclusive in the brain. And so the idea behind that is so that way we can make sure that the GP120 viral envelope protein is going to be transcribed and translated in the brain, and in this case, coming from the astrocytes. Okay, so that's a little just on the HIV mouse model that we're using. So it's just driven primarily by the GP120 protein. Then I'll touch on a little bit of what interferon beta is. So when the HIV virus makes its way, it binds to the surface of the cell, it'll release certain, it's a single-stranded RNA, so it'll release those components into the cytoplasm of the cell. And in the cytoplasm, this is a really simplified schematic, but there's a lot of innate viral sensors. So things that will not detect something specific for HIV, but will detect any viral RNA or viral proteins. Some of them you might have heard like TLRs, TLR, 7, 8, 4, which is on the membrane. But there's a few, and they'll signal downstream to the cell to alert it, essentially to activate a type 1 interferon response. And this is an innate response that will, no matter what virus enters, will initiate the activation of a series of different proteins, which will function to essentially try to mitigate the viral infection and alert neighboring cells that a viral infection occur. So the two of the most pivotal players in this are these IRF3 and IRF7, which after this cascade get phosphorylated and then translocate into the nucleus here to actually begin the activation of and increase the transcription of a lot of these type 1 interferon. And so when that happens, what you get is the activation of type 1 interferon alpha and type 1 interferon beta. And these work in a paracrine and autocrine-like fashion. And so it'll come back and it'll signal onto the same cell that it was synthesized from, but it could also be secreted and then signal to neighboring cells. And the idea here is to prime those neighboring cells for an antiviral response, because it's alerting them that the virus exists in this cell here. So everyone around me, heads up, there's a virus around. And when it signals, it creates this complex here, and that complex comes and essentially drives the activation of a few hundred antiviral genes and proteins. And the really cool thing with interferon beta is it's already approved treatment. It's used for treatment in RRMS. Um, and so it's a particularly um, viable option for a treatment for something like this. And by the way, if anyone has any questions along the way, like feel free to chime in partway through or at the end, whichever one you prefer. So previous studies in our lab took a look essentially at just whole brain between wild type mice and these GP120 mice, which really recapitulate a lot of the HIV symptoms in the mouse model. And we wanted to take a look at interferon beta just to see what's it doing. There should be an interferon beta response. There's a viral protein that's around, and you can see here that at 1.5 months, at least at the RNA level, we see about a threefold increase in interferon beta. But following at different time points, 3 months and 6 months, there's actually a return back down to baseline of interferon beta. And what you can see when you look phenotypically at these mice is at 1.5 months, we actually don't see much neuropathology. So there's no damage to the dendrites. There's no damage to any of the presynaptic terminals. There's no activation or little activation of astrocytes and microglia. But that really kicks in starting at 3 months and onwards. And so the previous student in the lab, Victoria, presumed that, well, we saw this return back to baseline of the interferon beta. So maybe the interferon beta is there to protect the neurons and protect the glia that are in the brain. And when it comes back down to baseline is when we start seeing all these neuropathological signs. So why don't we treat the animals with recombinant interferon beta? We treat them intranasally after the 1.5 month mark, and then we see if it helps neuroprotect them at the 3 month mark. And it was a 4-week treatment. And long story short, what they found was these interferon beta treated animals actually did help rescue the loss of the dendrites and the presynaptic terminals in the HIV GP120 mice. So we kind of have this idea now that interferon beta seems to be neuroprotective, at least in this context. And where my project kicks off is really trying to understand what molecularly and cellularly interferon beta is even doing in the context of the brain and in the context of our GP120 animals. And so here's like the first piece of data, which is looking at IRF3 transcripts. And you can see here that, again, there was these two pivotal proteins that were involved in initiating this type 1 interferon response. So wanted to parse out exactly what each of them were doing and how important they were in the various phenotypes that we have. So you can see that in our equivalent to HIV mice, we see an induction of this IRF3 at the RNA level. But when we knock out interferon beta, and this is like a global interferon beta knockout, we don't see any changes. And if you knock out the interferon beta from the GP120 animals, you also see there's no change in IRF3. So you can see that compared to GP120, the knockout with GP120 is the same, and compared to wild type and the interferon beta knockout, it's the same. Now, when we take a look at IRF7, we see here that one, there's a lot more of a robust upregulation from the GP120 animals. So it seems like HIV, the presence of the GP120 on HIV is driving this really pivotal antiviral player in the cell, or at least in the brain here. This is in cortex. But you can see here, when you knock out interferon beta in the GP120 animal, you get a huge return back down to baseline for a 4-star significance from the GP120 down here. So we know from this that interferon beta seems to be one of the key regulators of IRF7 and IRF7 expression. So we were lucky to actually be working with the NNTC, which is the National NeuroAIDS Tissue Consortium. And we got about 150 samples from patients. This is RNA from the neocortex. So we're able to run, and they're very well, the patient is de-identified, but there's a lot of clinical data that comes with each of those patients. And so wanted to recapitulate some of the things we were seeing in the mouse model with IRF7, and just make sure that that's what we see in HIV patients as well. So you can see here that after, again, running some qPCR, just look at the expression of IRF7, uh, compared to non-infected individuals, individuals with HIV, again, these are samples from the cortex, individuals with HIV actually have an upregulation of IRF7. And this is very similar to what we see here in our GP120 animal. And this is done with, I think, about close to 100 patients, 46 non-infected and 63 um, with HIV. And then what we can actually do is parse the HIV group even further. And so this is HIV patients that don't have brain pathology, uh, as seen via pathological assessment, to see if there's any like microglial nodules or multinucleated giant cells, versus HIV positive patients that do have brain pathology. And you can see here that primarily those HIV patients with brain pathology that have this upregulation of the IRF7 antiviral antiviral response gene. So it's trying to push back against the viral pathology that's there. And so so IRF7 was this kind of starting point for us when we looked at these patients. And we ran a few other genes that we were interested in just to see what was going on in a lot of the HIV positive patients versus the non-infected ones. And what kept showing up was this really strong correlation between um, what's called Ephrin-B1 and IRF7. And you can see here that although there's a subset of people who both don't have IRF7 expression and don't have Ephrin-B1 expression, the most prominent expressors of both of those things are those with HIV brain pathology. And if you parse it by actual, rather than brain pathology, which again is a histological examination, if you look at it from a neurocognitive examination, so do these people have behavioral symptoms that are behavioral deficits? You see that these same people who have brain pathology also have some form of hand, whether that be a mild form or a more severe form. And again, you have this really strong, at least for human data, strong correlation between the two. And so that opened up the floodgates for my research um, on Ephrin. So what's Ephrin-B1? What's it doing in the brain? What's it doing natively? What's it doing pathologically? Um, what cells is it on, and what's it doing to those cells? So those are some of the questions that I'd like to probably answer in the next 20 to 30 minutes here. But there's a really unique thing about these ephrins, which is um, they're actually so that they're usually cell contact dependent. So the ephrin is anchored onto one cell, and then the receptor is on another cell, and the cells need to be in fairly close proximity for the signaling to occur. Um, there's exceptions where they can be cleaved and then secreted and soluble, but for the most part, cell contact. And the point of this is really cool. Whereas in a traditional signaling system, you would see that the ligand will signal through the receptor, and you'll have all these downstream kind of effector functions. In this system, you have bidirectional signaling. So the receptor can in turn signal to the ligand expressing cell. And then there are scaffold proteins and effector proteins that come in here and can change the function of the ligand expressing cells. They call this forward signaling and then reverse signaling. And some of these functions include like cell migration and cell adhesion and repulsion, as you can imagine from like the close proximity they need to be in. And a lot of studies in development around neurons, so synaptic plasticity, and then a few lesser described ones around inflammation. And actually, one of the co-sponsors for my grant and one of the people on my committee actually works on Ephrin-B1 in the context of another glia, astrocytes, and they found that the expression of Ephrin-B1 helped drive the engulfment of neurons more, stemming from the astrocytes. So the direction that I wanted to take in my research was mainly to look at what's going on with inflammation and then what's going on with the ability to um, essentially engulf the neurons and what might the efferents be up to in the context of HIV. So uh, over here, so what effect does interferon beta elicit on microglia? And so particularly in the context of the ephrins. So really focusing my attention on microglia and really focusing my attention on the ephrin system here to see how interferon beta might be regulating that, because actually we don't quite know very well what's regulating the ephrins. So my hypothesis is that interferon beta confers neuroprotection in this HIV neuronal damage model, mediated in part by its effects on microglial Ephrin-B1. So I'll first take a stab at what are some of these regulators. And so here, using a public data set, was taking a look between HIV encephalitis patients and control patients. And you can see here that there's an induction of Ephrin-B1 and its receptor, EphB2, in HIV encephalitis patients versus control. So you can see this upregulation. And so that kind of validated, my intrigue in wanting to look at this. But you can also see, which is not so surprising, all these synaptic transmission, excitatory, and inhibitory neuronal marker genes, all downregulated in the HIV encephalitis group. So I went to take a look back at the NNTC data, and I wanted to take a look at EphB2. So again, this is, to give a visual, this is one of the receptors here. And like I said, the receptor can be signaled on from the ligand, but it can also reverse signal to the ligand to do other functions over here. So I went and took a look at EphB2 and just jumped to the part where we parsed through the brain pathology and no brain pathology. And you can see a similar profile to that of IRF7, which is, it seemed like in the brain of patients with HIV that have brain pathology, we see this upregulation of this EphB2 gene over here. And just to note, this elevated expression in the brain of this has been associated with different, inflammatory markers, like really common ones, TNF alpha and IL-6, but also in the context of like traumatic brain injuries in certain regions of those injuries. And when they ablate and get rid of EphB2, they actually see a return back to baseline of a lot of those inflammatory markers. And so this gave me the insight of, maybe the Ephrin system is a lot more important in HIV and in inducing the inflammation than we previously expected. And so this was taking a look here in the, again, the HIV transgenic model that we have, as well as some of those interferon beta treated animals, just to see, okay, is there an induction at the mouse level of Ephrin in either of these animals? And so you can see here, I'll direct your attention to the total segment, which is that this was done in the hippocampus, but we also saw the same thing in the cortex, which is it looks like the presence of this viral protein actually does drive Ephrin-B1 protein expression in the hippocampus. And it looks like the interferon beta treated animals are also driving expression of Ephrin-B1. And if you just gate around the microglia, which you can see here are in red, So this is a zoom in of it. You can see these green puncta. So the microglia seem to be expressing Ephrin-B1 on the surface of them, which hasn't previously been disclosed. And on top of that, it looks like we found a few regulators of that might be stemming from an innate immune origin. So the interferon beta, as well as a viral envelope protein, GP120, that seem to be inducing expression of the Ephrin system in the brain. And so we went back to some of the knockout animals we have, and I'll try to simplify this by just saying that when you remove interferon beta, you actually see a downregulation of Ephrin-B1 and this is in the hippocampus as well, of Ephrin-B1 and its receptor EphB2. So it seems like it's pretty clear that that interferon beta is regulating and GP120 are regulating this. And then the last kind of little puzzle piece in this story and this segment of the story is that when we take a look in vitro, so not in any mouse model, we take a look at microglial cell line HMC3. This is just to confirm that they express canonical microglial markers. But you can see that although we don't see it in the microglia in vitro with just GP120 recombinant protein treatment, with interferon beta, we actually do see an induction of both the Ephrin-B1 and the receptor at the RNA level in the microglia too. So we know here, so some of the conclusions are, so HIV neuronal damage is mediated primarily by microglia. Again, they're not focal point where everything else stems from. Interferon beta and GP120 drive expression of Ephrin-B1, which is the ligand, as well as its receptor, EphB2. And we can see that in the interferon beta treated animals, as well as those animals that have interferon beta knocked out of them. And we saw that in at the RNA and the protein level. And I showed mostly hippocampus, but we do see the same thing in the cortex. And like I said, ablating interferon beta reduces the expression of these. And just simply the fact that Ephrin-B1 exists on the microglia. And we saw that in those, the immunofluorescent images from the section tissue, that the Ephrin-B1 puncta are on the microglia. And it seems like they are being regulated on the microglia when you have GP120 or interferon beta. And so the next step I wanted to do was, okay, that protein is being expressed on microglia. What is it doing? And so to do that, we went to an in vitro model where we took the HMC3 microglia. So these are human microglia lines. And I did an siRNA knockdown to knock down the expression of Ephrin-B1. And then following confirmation of that knockdown, I ended up treating those cells with a recombinant EphB2. Again, this is the receptor that reverse signals to the ligand. And I want to see, when you reverse signal, what are the microglia going to do? And so first, I needed to double check to see, is my siRNA knockdown working? So you can see here that when I just treat it with the recombinant protein, there's no change in Ephrin-B1 transcript levels. But when I treat it with the siRNA, we see about close to a 60% decrease in expression of Ephrin-B1. And then in the treated, the EphB2 treated and the knockdown, we also see that same level of knockdown occurring. And then also confirm this at the protein level. So running a Western blot, you're able to see that essentially decreased in the intensity of the band here for the siRNA knockdown. You can see that there's less Ephrin-B1 protein as well as less Ephrin-B1 transcript levels here. So that gives me confidence that whatever differences I'm seeing as I treat these cells are going to be differences that are real and that are attributed to the knockdown of this particular protein, Ephrin-B1. Okay, so this is done just through qPCR, and you can see here a common trend, which is when I give them the receptor and it reverse signals to the microglia, it seems like the microglia are upregulating expressions of pro-inflammatory markers. So things like IL-6, things like TNF alpha, common ones relevant to HIV, CCL4 and CCL5, as well as some MMPs, MMP9, and a really important long non-coding RNA that help promote HIV infection. So that's all upregulated. But you can see that when you remove Ephrin-B1 and then treat it with when you reduce Ephrin-B1 and treat it with that same receptor, it actually perpetuates the pro-inflammation. You can see here that there's either a trend or significance when it comes to comparison of column 2 to column 4. So that was a little confusing to me, because if the receptor was going to signal through this ligand and it's inducing inflammation, why is it that when I remove that ligand, there's even more inflammation? Your intuitive response would think that when you remove the ligand from there, there's nothing to signal to. And so you should have a decrease in inflammation. So that was a little bit of the opposite of what I was expecting and what my hypothesis was. And so I wanted to look at some other pro-inflammatory markers. So STAT3 is a very common one, looking to see if it gets phosphorylated or not. And in our mouse model, we do see that. So we see that our GP120 animals have a very strong upregulation of phosphorylated STAT3. And what I had seen and what's been described in some other papers is this strong correlation with Ephrin-B1 and phospho-STAT3 in these animals. So I figured, okay, let's take a look at the STAT3 expression after I treat these cells. And I need to collect more experiments for this. I only have 2 biological replicates, but I don't quite see very much changes when it comes to the expression of phospho-STAT3. So it looks like there's an upregulation in inflammatory markers like IL-6, TNF alpha, things like that, but not so much when it comes to STAT3. So the next step was, okay, great. I can see that there's inflammation occurring in the HMC3, the microglial cell. We get inflammation, and that apparently that inflammation is a little bit worse when we remove Ephrin-B1. So when I run those studies, I'm also collecting some of the supernatants from those cells. So that way I can take a look at what are some secreted factors that might be changing here. So there's a really cool multiplex system called the Legendplex, where you can, it's a bead-based system, and you can look at 13 different markers out of the supernatants of your cell line in your experiments. And what you can see here is when you look at, so these are pro-inflammatory chemokines. So chemokine is essentially a protein that helps pull in or guide inflammatory cells to a particular region. And so you can see here, when we treat the microglia with EphB2, so the receptor, and induce reverse signaling, you see an upregulation of all of these pro-inflammation chemokines. So essentially what this means is, the gist of this is, when you signal to the microglia secrete these factors, and they're telling a bunch of inflammatory cells, hey, start coming to this region. We need you. And that aberrant activation would cause inflammatory cells to enter the brain here and just constantly be in the location, secreting pro-inflammatory cytokines, causing damage. And so you can see that for the most part, the Ephrin-B1 knockdown doesn't really change anything from column 2 to 4. There's some that trend downward. You see trends downward, and then some that are significant. So from this, we can see that reverse signaling by the EphB2 on microglia induces a pro-inflammatory cytokine and chemokine profile. again, in column 2, you can see that it was upregulated. Now, when you knock down microglial Ephrin-B1, for the cytokines, it seemed to actually enhance the amount of pro-inflammatory cytokines. But what was interesting was it almost gave the opposite effect when it came to the pro-inflammatory chemokines, where we started seeing those trends or those significance of a downregulation, um, in some of those markers as seen over here. Um, and so going forward, some of the last things that I'm going to be doing here to finish up will be want to recapitulate these findings with a better cell line. So these are an immortalized cell line I used, the HMC3s. And so I want to recapitulate these with iPSC derived microglia, which have, I think, a lot more appropriate of a phenotype for microglia, and then run an RNA-seq on some of these cells after treating them as I've been treating them, just to see what kind of other changes outside of inflammation we're seeing. And then a quick one over here is the last thing I wanted to look at as part of my project is looking at the impact of Ephrin-B1 on synaptic pruning. So again, in HIV, we see damage to the neurons. But what's really interesting is you don't see neuronal death. You just see an impairment of like the ability of those neurons to function. And so uh, presumably, and a lot of people have been describing this a lot more, is this capacity for microglia to prune away the neurons, which is very common in development, right? That's how it works. So you have the synapses, create more synapses than you need, and then the microglia are really active in pruning away the really weak ones. But when you have microglia that are very active, they can aberrantly just start pruning away synapses. And my hypothesis is that the microglia might be doing this in the context of HIV in the brain. And I think that Ephrin-B1 might be a pivotal player in that. And I say that because the Ephrin system can actually endocytose the receptor or the ligand from different cells. So it can, if it's a ligand expressing cell, it can engulf the receptors and chunks of the cell off of that. And so, like I said, there's also been a lot more in the field going into the fact that microglia can displace and prune away and engulf at phagocytose a lot of these neurons as well. Mostly described in development, but also now a lot more in pathology. And again, in our GP120 transgenic animals, as well as in human patients, they've done a lot of pathological studies on that. But you can see we're seeing a loss in synaptophysin and MAP2, which are presynaptic and postsynaptic markers. And so the idea here is, are the microglia aberrantly pruning the synapses of these neurons? And that's what's causing a lot of the neuronal damage we're seeing. So here I have a little bit of conflicting results. So I, again, ran the same study in the microglia, knock down the Ephrin, treat it with this receptor. And what I saw was this very common marker for phagocytosis. So the ability to engulf another cell and actually see that, okay, with the EphB2, we do see an upregulation of this. So as if when there's the EphB2 is around, the microglia are going to be engulfing things more. And when you knock out Ephrin-B1 and then treat them with this receptor, it's significantly higher. A little bit, but the significance is there. And so this, according to this piece of data, suggests that we're going to probably see an upregulation of phagocytosis and engulfment coming from these microglia. Well, I wanted to also show this in some context outside of RNA. So I used a common, and not very common, but it's called pH rhoda. And what it is these bacterial, these bacteria that are labeled with this pH rhoda. And the way that this works is when it's at a like physiological normal pH, neutral pH, the pH rhoda doesn't fluoresce. But the moment it makes its way into lower pH environment, so like the endosome or the lysosome, which is found in microglia after they engulf cells, it should fluoresce. And so I wanted to use this to see, okay, are the microglia actually engulfing more or less depending on the treatments that we gave them? And so, long story short, um, what I found was contradicting to some of the CD68 expression. So what I found here was that the EphB2 um, doesn't seem to be making any difference in the capacity of these microglia to engulf things. But when you remove Ephrin-B1, we actually see a downregulation in their ability to engulf these bacterial particles that are coming in. And so the jury's still out, I think, on the pruning side of things. Does removal of Ephrin-B1 and does the treatment with EphB2 have any impact on the capacity of these microglia to prune uh, and to prune neurons? And so my future studies on that portion are going to be to, I think, look at synaptosomes, which are like pieces that are secreted out of neurons, to see, and I can label those and see if the microglia are going to engulf those more or less. So I think that will be a next step when it comes to some of the phagocytic activities. And something I've been doing on the side is developing iPSC derived cells. And you can see here, you have the iPSC, the iPSCs, you form these embryoid bodies first, and then these neural rosettes, and then you form neural progenitors. And I'm now on the last step where I can actually differentiate these neural progenitors into either neurons and astrocytes. And I can see what impact the microglia are now going to have directly on the neuron. So it's a functional output to say, okay, I see all these inflammatory things changing. Do any of those inflammatory things have any bearing on the actual survival, the function, or the activity of the neurons? And that's where I'm spending the last 6 months here, trying to tackle. Uh, and so just to wrap it all up, HIV neuronal damage is mediated primarily by microglia, which is why we focused on those. Interferon beta and GP120 regulate Ephrin-B1 and its receptor here at the RNA and protein level in the brain. And Ephrin-B1 does exist on microglia, which actually hasn't been previously reported. And then when you reverse signal with the EphB2 onto the microglia, it induces a pro-inflammatory cytokine and chemokine profile. But when you knock down microglial Ephrin-B1, it enhances the pro-inflammatory cytokines, but seems to trend towards decreasing the pro-inflammatory chemokines secreted by those microglia. And EphB2 signaling, like I showed, induces this phagocytic marker of CD68. And when you knock down microglial Ephrin-B1, it actually made that a little bit worse, as if it would engulf the neurons a little bit more. But on the other side of the coin, I have the other piece of data with the pH-roto suggesting that actually the Ephrin-B1 knockdown seems to maybe alleviate some phagocytic activity coming from microglia. So to give it all in visual form, you have the microglia and they express Ephrin-B1. And you have other cells in the brain. So I'm using the neurons in this example that express the receptors. So when you have constant expression of these two things, when the microglia are activated or when you're in HIV, what you're seeing is an increase in synaptic pruning. You're seeing an increase in pro-inflammatory cytokine production and an increase in pro-inflammatory chemokine production. And what that does is it seems to damage the synapses around the neurons and makes the neurons not function well, leading to a lot of the behavioral problems. Now, when you remove Ephrin-B1, what we end up seeing is we see more pro-inflammatory cytokines. We see less pro-inflammatory chemokines that are being secreted, and the jury's still out on what's really happening with synaptic pruning. I'll probably take a couple more experiments to see which direction that one leans in. A lot of times, what you see is RNA expression doesn't always translate into protein expression. In fact, sometimes you see the opposite, where because you have so much protein being expressed, the cell downregulates the transcript levels of that. So that way, because it doesn't need very much RNA to synthesize more protein, there's already so much protein. So some of the RNA data, I think in general, take with a grain of salt and make sure to confirm on the protein level. And then, yeah, just big acknowledgments to my lab and my committee members and all my funding sources. And then I will also take any questions if anyone's got them.
That was fascinating, Jeff. Can you hear me
Okay.
okay?
Yeah. I can hear you, no problem.
Yeah, no, that was really amazing. I guess one question I have is, we know that in HIV and its interaction with CD4 cells, there are these fusion inhibitors that you have a short time window to administer, which have a pretty good effect, like enfuvirtide. Do you have any ideas about whether they would prevent the interaction with microglia as well?
Um, So the way that like HIV even makes it to the brain is there's like essentially they get trafficked up there. So it's called the Trojan horse method, where they infect the T cells and they infect the macrophages in the periphery first, and then usually through the macrophages they make their way through the blood brain barrier and then into the brain where they then infect the microglia. That's a primary way of doing it. Sometimes the virus can seep through the blood-brain barrier, um, but if if you can stop the and that happens, I know within a few days you'll see it in the brain after infection. They've done that in like a lot of non-human primate models. So I think if it's quick enough where you can prevent it from getting into the trafficked cells that make their way to the brain, um, then then it should be possible. Uh, then it should be possible to prevent it with those. So I don't see why it wouldn't work for the macrophage as well.
Thank you very much. Yeah, that's great. If there aren't any other questions, I can ask another one.
Fire
Um,
away.
so another question I had is like, it seems from the mechanisms that you demonstrated that once the interferon beta pathway has been activated and you get this recruitment of the inflammatory response almost, it's like from what you're saying, it seems like the horse has kind of bolted. Like we're now stimulating so many different pathways that perhaps have some interplasticity that actually, now we've got so many downstream reactions. Is this similar to other viral encephalitises, for instance, maybe herpes simplex, etc.?
It depends on, so it is similar. So like there's a lot of overlap actually with SARS-CoV-2. Again, like there's the spike protein and the spike protein, again, functions in like a pretty similar way where it can induce activation. Um, it a lot of it boils down to the, what downstream players are activated, and what type of virus it is. So this is in this context, it's a single-stranded RNA. You know, so very similar to some viruses, but there's other ones that are double-stranded and things like that. And what that the difference there is which viral detector detects them, right? So there's different TLRs that detect double-stranded versus single-stranded, and because different ones detect that, the signaling that occurs downstream is different. And so you'll have a heterogeneous expression profile, which means a heterogeneous like cellular response, which means a heterogeneous pathological profile, which means a heterogeneous symptomatic profile. Um, Kind of simplified, but that would be the progression. So it just depends, I think, on the type of virus, what's being detected from the virus. Is it a viral protein that's getting pushed into the cytoplasm? Is it just detecting a surface protein? Um, so it just depends on the type of virus that it is, would help drive the different response.
So with the similarities with the SARS-CoV-2 virus, I mean, could we do a similar platform phase 3 approach that we did where we try and repurpose a bunch of, antiviral drugs or dexamethasone, et cetera, and just see what the effects are clinically if we know that they're safe?
Yeah, yeah, I think so. So, for example, interferon beta has already been used for treatment for RRMS, but there was a study that looked at interferon beta for treatment of SARS-CoV-2. And if I remember the study correctly, I think it didn't help in terms of um, whether or not like the viral where it ultimately helped is like how quickly somebody was able to get out of the ICU. So if they were in like very critical health, they were able to um, bounce back from that a lot quicker and be out of the bedside with I can't remember what it was, but many days earlier to a week earlier than what they got without interferon beta. So I think there's something there. Um, whether or not that's just to mitigate the severity of the disease or to help clinically, or to just minimize the inflammation. I think that's uncertain, but it seems like there's at least something once they're in the hospital that's beneficial about the interferon beta treatment.
Yeah, that sounds really similar to the recovery results for COVID as well. So it's interesting there's that consistency. Thank you very
Oh,
much, Joe. That was great.
yeah, no problem. Okay. Yeah, if there's no other questions, thanks everyone for listening. Yeah, really appreciate it. And yeah, if you have any other questions, just feel free to DM me. I'm always happy to talk about this stuff.